Hi everyone, today we're moving on to macromolecules. So macromolecules are large biological molecules that exist within our cells, and they are essentially those molecules that are based on a carbon backbone. So many carbons strung together into building these large biological molecules. And they're important because they drive how the cell works. So things like proteins are built within the cell and they allow the cell to function in a particular dynamic way. Proteins are also utilized by the cell and broken down. And so these macromolecules are really important to the appropriate functioning of cells. So firstly, let's look at why carbon is the backbone of these macromolecules. So the first slide I have here is on carbon's importance. So if you take a look at my bullet points, carbon is important because if you remember back to chemistry, carbons have 6 electrons and actually 4 in their outermost or valence shell. Because they have 4 electrons in their outermost shell, they can actually form 4 bonds, which makes carbon a very special atom. So because it can form 4 bonds, it can build chains that are long or short. And so we have different types of carbohydrates that have long chains and some carbohydrates that are simple sugars and have short chains or ring structures. And so because carbon is so versatile, that makes it a really good backbone for building these macromolecules. As I just mentioned, rings or linear structures are the types of structures that can be formed through stringing together carbons. Carbon chains can also be branched or unbranched, meaning it can be one long chain that doesn't have any little chains stemming off of it, or it can have little chains stemming off of it. Carbons can also form single, double, or triple bonds. So not only can we have a single bond between 2 carbons, but remember carbon can actually form 4 bonds. So it can use another one of those bonds to form a double bond between 2 carbons or other atoms. And so because it can have versatile number of bonds, that makes it a really special atom as well. It can also bind to things called functional groups. And over here on the right-hand side of this PowerPoint, you have this table of different functional groups. And so if you look at this table, the properties are not quite as important as looking at the structure of the functional group because that really gives you an insight into the properties. So these are the most common functional groups that carbons will attach to. And so if you have a long chain, that is designated as R. So R stands for a long chain of carbons. And then your last carbon would then be attached to an oxygen and a hydrogen. So an O with an H, which is sometimes written as just OH, like you can see down here in this functional group. But here it's showing you the line. Now, it's important to know how many bonds oxygen can form. And now because oxygen is missing 2 electrons from its outermost shell, it can form 2 bonds. Now, this R group doesn't show the complexity of that carbon chains, but we know that this carbon that would be here is actually attached to 3 other things. Here it's just showing the functional group. So the carbon that's attached to an oxygen, which is attached to a hydrogen, and again, hydrogen only forms 1 bond, that's called a hydroxyl group. So wherever you have an OH, we call that a hydroxyl, and that's an alcohol group. It bestows polar properties upon that molecule. A methyl group is just a carbon bonded to 3 hydrogens, and it's nonpolar, so we have equal sharing. A carbonyl is where you have a carbon in the middle of 2 more spanning carbon chains. So here this R group on the left represents a long carbon chain. This R group on the right represents a long carbon chain. And so a middle carbon, when it's double bonded to an oxygen, we call that a carbonyl group. And that again bestows polar properties upon that molecule. We won't see the carbonyl too often. Mostly we'll see the hydroxyl group, and the next one, the carboxyl. So a carboxyl is where you have a long string of carbons, and then that long string of carbons is attached to the end carbon, so one more carbon. And then that end carbon is attached to a double bond to an O, oxygen, as well as a bond to an oxygen, which is bonded to a hydrogen. So again, this is a hydroxyl. But when you put a hydroxyl together with a carbon double bonded to an oxygen, these together make a carboxyl group, and it bestows acidic properties upon that particular molecule. The next one, amino, we see these in amino acids quite a bit. So here we have a long carbon chain represented by the R. We have a nitrogen group, and the nitrogen is bonded to two separate hydrogens. And nitrogen has 3 electron missing in its valence shell. So it wants to make 3 bonds. So that's why you see 3 things bonded to nitrogen here. And so when we have 2 hydrogens with a nitrogen at the end of a chain, that is an amino group, and it bestows basic properties upon that particular molecule. Phosphate groups, again, just important to recognize these groups as phosphate. We have a central phosphorus atom, and it's bonded to, it makes 5 bonds actually, and it's bonded to anything containing oxygens. So here we see oxygens and OH groups, and we call that a phosphate, and that begins to be important when we discuss DNA. And this one you won't see much in our discussion, but it is a sulfhydryl, so sulfur bonded to hydrogen. So how do we build and break down these large biological molecules called macromolecules? Another way to refer to these macromolecules is polymers. And we create or build polymers in the cell by connecting smaller pieces together, and the smaller pieces are called monomers. So monomers are your building blocks for larger molecules called polymers. And so we call them monomers and polymers because that is a more general term used for building these large biological molecules. So we have carbohydrates, lipids, proteins, and nucleic acids, and they all have different names for their building blocks that you put together and build into the larger form. And that's why we just use the general term polymer and monomer. So dehydration is the term for building these polymers, so building carbs, building lipids, by connecting smaller pieces called monomers together. So this is a picture straight from your book. Here we have a couple ring structures based on carbon chains, and you can see various functional groups there. Now we have one here and one here, and this is a chemical equation. There's a plus in between, and then we have an arrow. So what this indicates is these are our reactants, these monomers that we're putting together to build something larger. And here's our larger molecule over on the right side of the arrow. You can see it's connected by this oxygen group. So we actually remove an OH from the first one and an H from the second one, and these red letters put together make H2O. So we take away water, hence the term dehydration, and we are able to put these monomers together into a larger polymer. So we release water when we do this. Now, on the other hand, if we want to if we're eating, for example, and we take in a carbohydrate, we want to break it down. So breaking down these larger polymers is called hydrolysis, and that's where you break them down into smaller forms called monomers. And this involves using water instead of releasing water. So it's essentially the opposite. Here on the left-hand side, this is now a different reactant. This is what we want to break down instead of build. So we want to break this down. We need to add some water. Remember, all of life's chemical reactions happen in water. So our cells contain water. So we want to break this carbohydrate down. We add water, and that will help to break the bonds between these two monomers. And you can see that arrow again shows you that we now have products upon the breaking down of this polymer. And we have our two short monomers, and we see a plus in between to indicate that these are our products that are released and now they are broken. And so an OH group is added back onto the first one and a hydrogen group is added back onto the second one through water. Now again, those 4 macromolecules are listed in this table that I made for you. So here on the left-hand side, we have the 4 macromolecules: carbohydrates, lipids, proteins, and nucleic acids. So these are the functional macromolecules of the cell. Here in these columns, I have their basic structure, examples, and functions. And it would be important to read the material pertaining to each one of these in your textbook. Briefly, I'll summarize each one of them. So carbohydrates, your basic structures of carbs, and they're usually long chains of carbons put together, and the carbons are usually ring form, so circular. They are made up of simple sugars. So carbohydrates are all sugars. You can, if you've heard the terminology monosaccharides, disaccharides, and polysaccharides, that's the term given to how many of those sugars you have put together. So if we go back here to this slide, one of these rings, that would be a monosaccharide. When you put 2 of them together into this structure here, that's a disaccharide. And when you have many of them together, that's a polysaccharide. So glucose and fructose are a good example of monosaccharides. Sucrose or lactose would be a good example of a disaccharide. And polysaccharides, some examples include glycogen, starch, cellulose, and chitin. And most of their function is for energy. So in fact, you could answer energy to each of these macromolecules and it would be correct because they are all used for energy. Lipids is the next group of macromolecules, and lipids are based on a fatty acid structure. So fatty acids, for example, triglycerides, written here under examples, includes a glycerol group with 3 fatty acid tails. That is an example of a type of lipid. Saturated and unsaturated fats, if you've heard of these before in nutrition classes, they are types of lipids as well. Steroids, so for example, cholesterol. Cholesterol is a type of steroid that gets converted into testosterone and estrogen in the body, so it's really important. It's based on a 4-ring structure, and it is a type of lipid as well. Waxes are another example, and phospholipids are yet another example. And phospholipids, you'll see an image of these when we talk about the cell. Phospholipids make up the cell membrane, which is the outer part of the cell. So phospholipids are actually phosphate groups, so a phosphorus attached to oxygen in a circular form, a phosphate group attached to 2 fatty acid tails. And so this phosphate group attached to 2 fatty acid tails makes up a phospholipid. And phospholipids make up the outside of a cell. And in fact, they're arranged in a bilayer, so a double layer surrounding the cell. Now, the fatty acid tails in phospholipids and even in triglycerides, they do not like water, so they are called hydrophobic. Whereas the phospholipid head in a phospholipid actually does like water, and it's called hydrophilic. And so you can imagine if there's water inside of a cell and outside of our cells there's water as well, what happens is if you try to arrange phospholipids in a bilayer around the cell to support the cell, your phospholipid tails would have to face towards each other, and the phospholipid heads would actually point into the cell and out of the cell, so in opposite directions. Now, because phospholipids make up the cell membranes, that's why I wrote here one of the functions is to provide cell membrane structure, but some of the other examples there are useful for energy as well, like triglycerides, saturated and unsaturated fats. Now, going back to the definitions of saturated and unsaturated fats, all that pertains to is carbon-carbon double bonding. So what saturated means is that the carbon-carbon chain is saturated, meaning it has the maximum number of hydrogens surrounding it. That means there are no carbon-carbon double bonds. Unsaturated means there is not the maximum number of hydrogens surrounding the carbon chain, and hence there must be carbon-carbon double bonds within the chain. So unsaturated fats do have carbon-carbon double bonds. Proteins are the next group of macromolecules, and they're based on amino acid structures. And I wanted to skip just a little bit to the next slide and show you an amino acid structure. So an amino acid structure is based on a central carbon. We have 20 amino acids that build up the human body into proteins. And so each one of those has a central carbon atom that's attached to 3 consistent things. So remember, carbon can make 4 bonds, but it is attached to 3 things that never change amongst those 20 amino acids. The first is a hydrogen group. The second is called a carboxyl group. You learned this in our first slide on functional groups. This is a carbon that would be double bonded to an oxygen, and the carbon would also be bonded to an OH group. And how that's actually written is COOH to indicate that functional group. And then on the opposite end, it's bound to an amino group, so nitrogen with 2 hydrogens. And then the fourth group that carbon is bound to is called an R group. And R stands for radical or side group, and it can be anything. So 20 amino acids that make up the proteins in our body, they all have different fourth groups. And so we just indicate that by the letter R to signify that this could be anything in there that makes up the particular amino acid. So R will differ amongst the 20 amino acids. Now, amino acids build up proteins, and some examples of proteins are hemoglobin, insulin, and enzymes. However, proteins do everything in the body. So there are proteins involved in transport, proteins involved in cell signaling, cell communication. And so for any function in the body, you have proteins that do or perform that function. And so they are generally just termed by the function in which they're involved. And so the functions are really any function within the cell, and it includes providing energy to the cell as well. The fourth group of macromolecules are nucleic acids. And nucleic acids are based on a nucleotide structure. And so a nucleotide structure is on the next slide, so I'll just take you there to mention the structure. A nucleotide structure is based on a phosphate group attached to a sugar, which is also attached to a base. So it has those 3 components, phosphate, sugar, and base, and specifically called a nitrogenous base. So these 3 together make up a nucleotide. So this should really say, instead of nucleic acid, nucleotide. And so a phosphate, again, is a phosphorus with oxygen surrounding it. A sugar is either ribose for RNA, which is an example of a nucleic acid, or deoxyribose for DNA, which again is an example of a nucleic acid. And then here, the base is actually a nitrogenous base. So if you've heard of the bases for DNA and RNA before, for DNA, they're A, C, G, and T, adenine, guanine, cytosine, and thymine. And if you're RNA instead, the bases that make up RNA are A, C, G, and U. So adenine, guanine, cytosine, and uracil. And we'll talk much more about that when we discuss DNA and RNA later on. And so just generally, those are the 3 components that make up nucleotides, which are the building blocks. When you put together a bunch of nucleotides and you string them together, you make nucleic acids like DNA and RNA. So DNA and RNA are long strands of several of these nucleotides put together. ATP is also an example of a nucleic acid. It has a really similar structure to DNA and RNA. And ATP is adenine triphosphate, and it's the main energy source of the cell. And we'll talk more about it when we discuss cellular respiration. So in terms of functions, energy is of course one of them, but also for DNA, it carries the genetic information for the cell. So that's a really important function of nucleic acids. And so that ends our discussion on macromolecules. Thank you for listening.